right now, one of the standards of care in the treatment of localized prostate cancer is the combination of radiation and androgen deprivation therapy. People sometimes call it hormone therapy. Hormone therapy has many side effects, and although it has been shown, we've showed in a MARCAP meta-analysis that it significantly improves survival in an unselected population of patients, it's very clear that not all patients derive benefit from it. And the side effects, because it lowers testosterone, are bothersome to many men: decreased libido, hot flashes, weight gain, loss of muscle mass. And so there's been a desire for many years to identify predictive biomarkers to be able to identify which men should or should not have hormone therapy. However, there's been no validated predictive biomarkers to do so. So instead, we rely on prognostic biomarkers to identify patients that have a very low risk of recurrence, to have sort of a shared decision making with patients to the pros and cons of receiving hormone therapy. So, we're sort of in an era now that, due to computational power and kind of the era of artificial intelligence and deep learning, that we can now go far beyond our typical variables in prostate cancer, PSA, Gleason score, by capturing a wealth of unused data. In this study, I'll be focused on the actual histopathology slides that right now are only used for Gleason grading by a pathologist, but there's a wealth of information. And so we had the hypothesis that using 5 phase 3 randomized trials, that we could go back and get all of their histopathology slides, digitize them, and using AI technology to be able to create and train a model and validate it to potentially be the first predictive biomarker to be able to offer to patients who should or should not receive hormone therapy. So what was done is, we worked with the NRG Oncology. They're a NCI-operated clinical trials group in primarily North America. And we got access to 5 randomized phase 3 trials that treated patients with radiation or with radiation and various durations of hormone therapy. We accessed the NRG Biobank to actually access all the slides, and we had them digitized so that they could now be sort of in a quantitative state that can then be fed through a series of novel pipelines that a team of artificial intelligence scientists that we worked with at a company called Artera, and using both the clinical data, right, their age, their Gleason score, T-stage, PSA, as well as processing all these slides through and having feature extractions. And right, these were non-annotated slides, meaning that it wasn't that we were telling it to only look at certain features. It was sort of an unbiased, I guess you could say, screening of the slides to pull out relevant features across the thousands and thousands of samples. In total, there was about 16 terabytes of imagery data. And so 4 of the trials were used to train the model to predict differential benefit or differential outcomes in patients receiving radiation versus radiation plus hormone therapy. And the primary endpoint was distant metastasis, a very clinically meaningful endpoint. Once that model was optimized and locked, we then independently, with an entirely separate randomized trial, a separate biostatistician, we applied that locked model to RTOG 9408. It's the largest randomized trial, and it's very important because it's randomized men to radiation with or without short-term hormone therapy. And the vast majority of the patients on that trial had available slides to be imaged. And after applying, we then applied the model to that trial to look at what we call a biomarker treatment interaction, to see would there be differential benefit based on patients who are biomarker positive or biomarker negative from the receipt of hormone therapy. The key findings is number one, we were able to successfully train a model that was able to identify differential benefit of hormone therapy. And probably even more important than that, because you can train things to do a lot of things, is we independently validated for the first time ever in a randomized trial that this biomarker, this predictive AI-derived biomarker, predicted differential benefit from hormone therapy. And so about 1/3 of patients on that validation trial were biomarker positive, and they had a substantial benefit of hormone therapy, about 10%, with long-term rates of improvement in distant metastasis. Whereas biomarker negative patients, which was 2/3 of the patients on this trial, had basically the hazard ratio was 1.00, meaning there's effectively no benefit at all from the receipt of hormone therapy on metastasis. So this is very powerful, and we looked at other endpoints as well: death from prostate cancer, metastasis-free survival, and all of those endpoints also showed that there was significantly greater benefit of hormone therapy in the biomarker positive patients, and effectively no benefit in the biomarker negative patients. This is, a very rare project in the sense that, usually when you create a biomarker, it sometimes is many, many years till you have sufficient evidence to where you'd say it's clinically ready to be deployed. Our group combined with NRG and Artera and a very large team, learned from sort of prior biomarkers we've worked with to sort of go straight to high level, high quality randomized data. And, so what we have now is a predictive biomarker with independent validation in a randomized trial. And so this really is sort of the bar that most of the time is used to say that this is clinically ready for deployment. And so I think that in 2022, hopefully by the end, we'll be able to have this available for people to order, so where they can send their slides to be digitized and this algorithm run on their slides. And this is going to be profound for patients, because they can now, rather than kind of just telling all patients with predominantly what we call intermediate risk prostate cancer to receive hormone therapy, the majority of them can probably successfully omit hormone therapy with confidence that they will derive no intrinsic biological benefit. So I think this is going to be a major game changer, and I think this is really the this is the beginning of a whole new era of AI-derived biomarkers that are going to really help personalize therapy. There was almost 6,000 patients that enrolled on these trials. So really, we have to thank the patients' families for enrolling and participating in research, NRG Oncology, all of the investigators, collaborators, Artera. Really, it's a huge, huge team effort, and very exciting work.